Hi guys, just thought I'd better make that second chapter 7 podcast for you, and I'll do chapter 8, chapter 9, and a little sack podcast today as well, and get those up online for you. So, of course, we've already discussed pathogens causing disease. So let's get that started. Where are we? It doesn't want to do it. Come on, you can do it. No? No? Alright, we'll go slide by slide. We get around this. So, obviously we discussed what disease was and what the various pathogens are, and then there's a whole lot of things we can use to keep pathogens under control. So, disease. Now, of course, this whole last bit of the second area of study is really a case study in signal transduction. So, we started by looking at what homeostasis was, so that idea of maintaining a relatively constant internal environment. And dis-ease is when that relatively stable environment becomes unstable, or conditions change that we have to respond to. And these often are caused by infectious diseases, which are caused by pathogens, or through non-infectious means. You might inherit genetically the disease, you might have poor diet, poor exercise, have an accident, great deal of environmental stress in your home life or work life, or all those sort of things that can cause other problems. So dis-ease is our ability to maintain homeostatic control overwhelmed by something. So we then have a whole range of pathogens. We're going to look at infectious diseases in this part of the course. We have a whole lot of pathogens which will cause dis-ease. And generally speaking, it's not the pathogen itself, but rather its metabolic wastes which tend to create the problems. So we basically classify them in three groups. We have things like viruses here, which are non-living. And in that non-living group, we also have viroids and prions, which we'll talk about soon. We have the prokaryotic things like bacteria. So we have wonderful bacteria down here, which can be obviously do good things as well, and also things like worms and fungi and other living organisms that are multicellular rather than single cellular or small cellular bacteria. So, bacteria. Essentially, bacteria are prokaryotic. Well, they are prokaryotic by definition, sorry, which means, of course, they don't contain membrane-bound organelles. Membrane-bound organelles, I'll get that right yet. And they don't have DNA in a nucleus that tends to be free-floating. But they do have ribosomes, they do have RNA, they are able to self-replicate, and of course these prokaryotic cells pre-exist their own eukaryotic cells. And we've talked about endosymbiotic theory before. So these things have been around a long time. We classify them a number of ways, and you will have seen that video last week, the idea of the Gram stain. And Gram stain tends to give us a clue to which way to treat the bacterial infection. So one of the first things to do is to eliminate half of the different bacteria. But we can also look at their shape. And so we have circular bacteria called cocci. The long rod-like ones are bacillus. And then we have ones that are spiral-shaped. We call those spirochete. And so the shape also helps us identify the species. Identification is really important because it allows us to then look at what best treatment to use to destroy the bacteria and cause the patient some relief. They also organise themselves differently. Some are solo in their life form, so just single or small groups of cells. Others form huge colonies, and others form multicellular colonies of different bacteria and fungi. Some have capsules. Some, like this one in the picture here, is a lovely long flagella, as well as cilia around the outside, so it's quite mobile. Some aren't mobile at all. Some work best in anaerobic situations, some in aerobic, and they have different nutritional requirements as well. So all these things allow us to work out what bacteria we've got, and we can obviously treat that. Oops, went too far. So, particularly with bacteria, it's generally the toxins that they produce from their metabolism which causes us problems. So the bacteria essentially acting as a host on our body, a parasite on our body, we're the host, and using the fact that our interior is warm and wet and protected and there's plenty of food. So of course, in those situations, they're happy to work away, feeding happily, growing to large colonies, but then they start producing waste. And those wastes tend to be toxic to our body. And often those toxins are then taken up, especially from the gut flora, entering our bloodstream and causing us great difficulty. And some classic examples are tetanus, typhoid, and tuberculosis. The good thing about bacteria is they tend to respond really well to antibiotics, although, of course, as we discussed in class, antibiotics are becoming more and more difficult as we overuse them. We also have things called protozoans. Protozoans often are single cellular, but they also are pathogenic, so they live as parasites on us. Again, they'll produce toxins from their metabolism, which will cause us to feel sick if there's enough of them to produce a large dose of toxin. So as they live inside us, they enjoy our warmth and moisture and food, grow into large colonies, produce lots of toxin, and we become quite sick. Some are flagellated, so we have these guys with tails, and they're able to swim around. And a good example of that is, I always get these wrong, trypanosoma, which causes the classic sleeping sickness. Some are known as sporozoans, and they have little spores, obviously, they use for reproduction. And the plasmodiums, which we know cause malaria, are sporozoans. And sarcodians, such as amoebic dysentery, is another one, waterborne disease that gets passed around quite quickly. So the protozoans are quite an issue, and they have the thing with protozoans, they have quite a life cycle. And so if we can work out how best to break the life cycle, then we can actually treat the problem. And those of you guys who work in ag hort, you'll know that some of these things are quite common in sheep and cattle and other livestock. And again, one tries to treat the protozoan by breaking its life cycle. And yeah, that's probably enough of that. We talked about it in class, to me. Oomycetes used to be considered fungi, but they've been reclassified as protists, which in protists essentially they're miscellanea. And they have a presence of flagella, and they have a cellulose wall. Now, fungi don't have cellulose walls, so these guys are more closely related to the plant possibly than the fungus is. Fungi are probably more closely related to us than they are to plants. And these are generally plant pathogens. We don't know of any oomycetes which cause problems for animals, but that's often hard to tell because, as you can see from this little diagram here, there's a single oomycete. There's actually a Phytophthora cinnamoni, what used to be called cinnamon fungus. That's one little individual, and that's at about 1,000 times magnification. So they're really easy to pass around and move from site to site, and they're tiny and hard to pick up. So quite often these oomycetes will get passed around very quickly, and it's really difficult to control. Fungi, obviously, like the bacteria, there's lots of good fungi too. We eat lots of fungi. Fungi help us break down things in the environment, but sadly they also are well known for causing disease. And in our situation on humans, we know of tinea, ringworm and thrush, all three of which are caused by a fungi. But they're quite easily treated because, of course, we can put a topical treatment on the tinea or ringworms or on thrush and cause that fungal infection to die off and disappear. What we see here are the fruiting bodies. So most of the fungus is actually not seen. We only ever see the fungus when it comes into fruit, and this will now drop spores. And of course, at the moment with the rain finally coming and autumn still with us and a bit of warmth, we're going to start seeing lots of fungus under our trees and amongst the lawns and things as they start to start to fruit. And they'll be busy fruiting because they need to put that fruit out for next year's what am I trying to say? Next year's crop. We also have lots of worms. So worms are another major problem. As you can see, this hookworm here has wonderful hooks to latch onto its host, us, and while it holds on there, it then starts to take in all the food that is coming past, because these are found in our digestive system, as are these guys here. They're both well known for their living in digestive systems of mammals, and why wouldn't you? There's lots of food and warmth and protection, and they basically hook onto the side of the intestine and just suck your dry food and cause horrible problems. And of course, they also have a life cycle. So again, what you're trying to do is break the life cycle. And here's a whole lot of babies coming out the anus. So if we can wash hands and not pass these around, then you've got one way of breaking it. And again, you can use wormicides to kill these things. Quite easily killed. Oops. Arthropods, lots of different insects live on our skin. A lot of them are quite useful. There's little mites that live around your eyes, tiny microscopic little mites that clean up the mess around your eye. They're quite useful, really. And again, others are horribly damaging and cause all sorts of fevers and disease. But again, they're living on the outside of your body, so they tend to be easily treated. Viruses, however, are a bit more of a problem. A virus, of course, is a non-living thing, and they can stay in the environment for a long, long time. It's microscopic, they're tiny little things, hard to see under a light microscope. You need an electron microscope to find them. And they inject their DNA from their protein capsule down through here into the host, and the host, of course, replicates for them. Some of them contain DNA, others contain RNA. We call the RNA ones retroviruses, and they aren't able to replicate for themselves. They can only have DNA or RNA, and they don't have ribosomes. Ribosomes are also tiny, and some suggest may well be viruses themselves. So it's very hard to control, because again, they're tiny, they're long-living, and they're not alive. So you can't kill them, you can only sort of block their life cycle. And again, treatments now are trying to find ways of stopping this, injecting its DNA. And viruses attack both animals and plants. A lot of viral diseases in plants too. Viroids, interesting little things. Just a piece of RNA, sort of free-floating RNA, single-stranded, no protein coat, no membrane. It's not alive, so like the virus is non-living, but being RNA is capable of replicating. And they tend to affect plants. And a number of plant diseases, including the one that causes skeletonization of leaves, where these things enter and remove all the soft tissue. And of course, if you remove all that soft tissue with chlorophyll, then the plant has trouble photosynthesizing and getting energy, and they die. So a major problem for farmers. But there's not that many, but there's a few viroids. Prions, of course, we talked about in class 2, and prions are another really quite scary pathogen. Non-living, simply a piece of protein. And so it's free-floating protein. If we get the prions that we know of, if they get into the body, tend to attack the brain. So here is a diagrammatic form of Creutzfeldt-Jakob disease, or what we sometimes call mad cows. This is one in humans. Mad cows, better known as bovine spongy encephalitis. Both attack the brain and destroy the brain. And how do you control this? This is a piece of protein. How do you recognize it? Where do you find it? It's really difficult to recognize what's going on here. A number of diseases which have defied classification, people now look for prions as a possible cause. Don't always find them, but that's one way of trying to find a cause. And of course we can control these things. So, we use antiseptics on our skin to kill pathogens, we use disinfectants across objects to try and kill pathogens, and we use antibiotics, of course, to kill bacteria. So we have chemicals that we can use to control the spread and when they've attacked us, to actually reduce the infection quite quickly. We also have antimicrobials, fungicides, antivirals that are now being developed for many diseases, and we have chemicals for protozoans and helminths, the worms. So we are able to use chemicals to break life cycles, to clean surfaces, to stop spread, and then to kill off the pathogen when it arrives in the body. And the antivirals are one of those really exciting new areas where we might be able to control viruses when they attack people. I just made in under the 15 minutes. Well done me.